What is up, guys? It is your boy Zach here, aka That Bio Dude, coming at you live with the most informative, funniest, and coolest AP biology content that you can find on the interweb. And today we're coming at you with bio lecture number 8, sense organs. Let's get into it. Alright, so chemical senses. The sensory receptors for taste and smells are termed chemoreceptors because they are sensitive to certain chemical substances in food, including liquids and air. Chemoreception is found almost universally in animals and is therefore believed to be the most primitive sense. The location and sensitivity of chemoreceptors vary throughout the animal kingdom. They are very important in finding food, locating a mate, and detecting possible dangers. For example, in invertebrates, such as a frog, chemoreceptors are located in the nose, mouth, and skin. Alright, sense of taste will be the first one we'll touch on. In adult humans, approximately 30,000 taste buds are located on the tongue. Many taste buds lie along the walls of papillae, the small elevations on the tongue. There are also isolated taste buds on the hard plate, hard palate, pharynx, and epiglottis. There are 5 primary tastes: sweet, salty, sour, bitter, and umami, aka savory. Got some chips right here. That would be salty taste. Love me some Qdoba chips. Alright, so on the picture on the bottom here, you got your tongue, tonsils, epiglottis. Take a step further. You can see the papillae, those small regions of your tongue. If you look closely, you can see them. Even further are the taste buds on the papillae, and then you can break that down to one single taste bud there. That last picture, I thought it's pretty cool. It's got that sensory nerve fiber and the supporting cell and the taste cell connected to the microvilli that all work together with the connective tissue to help you taste food. Sense of smell. In humans, the sense of smell of olfaction is dependent on between 10 and 20 million olfactory cells. That's a lot. These structures are located within olfactory epithelium high on the roof of the nasal cavity. Here's how it works. So the olfactory epithelium in humans located high in the nasal cavity. Olfactory cells end in the cilia that bear receptor proteins for specific odor molecules. The cilia of each olfactory cell can bind to only one type of odor molecule. For example, if a rose causes olfactory cell sensitive to blue and green odor molecules to be stimulated, which you can see right here, these little odor molecules, the primary olfactory area of the cerebral cortex interprets the pattern of stimulation as the scent of the rose. And then you can see as it goes up through these olfactory epithelium, right up into the brain, the frontal lobe, and then break it down even further. There's the odor molecules going up into the sensory nerve fibers, up into the olfactory bulb, into the neurons in the brain, and this helps you determine what you're smelling and what it smells like, if you like the smell or if you don't. Sense of vision. Photoreceptors are sensory receptors that are sensitive to light. Some animals lack photoreceptors and depend on sense such as smelling and hearing instead. For example, moles live underground and use their sense of smell and touch rather than eyesight. Like in the movie In The Incredibles, at the very end, those moles say they're going to take over the world. I don't think they ever did. There might be a second Incredibles coming out. I don't know, but they couldn't see anything, so. Arthropods, compound eyes composed of many independent visual units called ommatidia. Insects have color vision, yet they make use of a slightly short range of electromagnetic spectrum compared to humans. However, they can see the longest of the UV rays, and this allows them to be very sensitive to particular parts of flowers, which is key to their survival. Got to get that pollen. Vertebrates have a camera-type eye. This is us. The lens changes shape to aid with focusing, and all the photoreceptors are similar to a piece of film in a camera. Yet the human eye is much more complex than this. Alright, here's a human eye up here on the right. I'll kind of talk about it. So we got the sclera, which is the outer white layer on the back and the sides of the eye. You have your choroid, which is the next layer, then the retina. These are the 3 layers of your eye. On the front, you have the cornea, which that's like the clear part that you can see, which you don't want to scratch that, you know. Then you have your pupil and your iris. Iris is the color. Pupil, which is the one thing that can dilate, a little black dot that you can see. And then you have the anterior compartment filled with the aqueous humor. And then you have the vitreous humor. Vitreous humor is the jelly-like one. The aqueous humor is water-like. And then you have your foveus centralis, which helps with focusing. And then you have your optic nerve and the retina blood vessels that help connect your eyes all the way back to the occipital lobe, which we talked about in the last video. And so, starting from the top, the sclera protects, supports the eyeball. Conjunctiva moistens eye surface. The cornea refracts light rays. The pupil emits light. Choroid absorbs stray light. Ciliary body holds lens in place. Iris regulates light entrance. The retina contains sensory receptors for sight. Rods make black and white vision possible. The cones make color vision possible. We'll talk a little bit more about rods and cones in a little bit. The fovea centralis makes acute vision possible. Lens refracts and focuses light. Humors transmit light rays, support eyeball, and then the optic nerve transmits the impulse to the occipital lobe and then to the brain. Alright, the focusing on the eyes. When we look at an object, light rays pass through the pupil are focused on the retina. The image is produced much smaller than the object because the light rays are bent, refracted, aka refracted, when they are brought into focus. Focusing starts at the cornea and continues as the rays pass through the lens and the humors. The image of the retina is inverted, it's actually upside down, and reversed from right to left. When this information from the retina reaches the brain, is processed, flipped over so we can perceive our surroundings in the correct orientation. So you have a picture up here on the right. You got the top one, we're focusing on a distant object. The light rays are coming in. The ciliary muscle was relaxed. The lens is flattened so you can see a little bit further. And the suspensory ligament is taut. And you can kind of see as the light rays come in here, they cross that fovea centralis in the back. And then focusing on a near object in the bottom, the ciliary muscle is contracted. The lens is rounded because it's closer to you, so you can get a better look. And the suspensory ligament is also is relaxed versus taut. This allows you to see that close-up object and focus on it better. And then you can see it kind of takes a bigger cross. The light rays cross a little bit bigger towards the fovea centralis. Distance vision. Here are a few disorders that force people to need glasses or contacts, like these glasses right here. Got them for $1. Pop the lenses out, look sweet. Okay. Nearsightedness. That's when you have trouble viewing distant objects. So they'll have a long eyeball, raised focus in front of retina when viewing distant objects. Concave lens allows subject to see the distant objects. So you need that concave lens, kind of picture cuts it off, but it needs to look something like that. Have that concave right there, which helps that nearsighted person see something far away. Farsightedness, right here in the middle. That's when you have a short eyeball, raised focus behind retina when viewing close objects. So you're going to need a convex lens that allows the subject to see closer objects. So you can kind of see the shape of that. And then astigmatism is an uneven cornea. Rays do not focus evenly. Even lens allows subject, an uneven lens, what you're going to need, allows the subject to see objects clarify. I'm pretty sure you can get work done on your eye too to get this fixed, but that seems painful. Alright, photoreceptors. Vision begins once light has been focused on the photoreceptor on the retina. The photoreceptors in the human eye are categorized as rods and cone cells. You can see that right there. You have, here's a rod cell, kind of shaped like a rectangular cylinder. And then you have a cone cell, which is shaped like a 3D cone, you know. The outer segment of rods and cones contains sacs of membranous discs, which contain visual pigments and rods. The membrane of each disc contains rhodopsin, a complex molecule containing the protein opsin and a pigmented retinol. When rhodopsin absorbs light energy, it splits, releasing opsium, which sets in motion a cascade of reactions that cause ion channels in the plasma membrane to close. Thereafter, nerve impulses go to the brain. And that's how rods and cones work. Very important to us in order to be able to see and perceive what we're seeing. Sense of hearing and balance. The ear has two sensory functions, hearing and balance, aka equilibrium. The sensory receptors for both of these are located in the inner ear, and each consists of hair cells with stereocilia, aka which is just long microvilli that are sensitive to a mechanical stimulation. They are mechanoreceptors. The human ear is separated into three main regions: outer, middle, and inner ear. Alright, so here's a picture of the human ear. You got the outer ear, which is this section, middle ear, this section, and the inner ear, which is this section right there. And I'm going to kind of talk about the pathway that it takes, like a sound wave coming from all the way from the outside of your ear, getting into the auditory tube to your brain, so you can understand what you're hearing and perceive it and then reply. So it starts in the pinna, which is right here, comes down through the auditory canal, through the tympanic membrane, which it then comes through these three little structures right here, the malleus, incus, and stapes, and then heads on over to the cochlea, which it then goes in this twisty guy right here, out the auditory tube, and then boom, right to your brain. And that's how sound comes from the start of the ear and gets to your brain. And then another picture we have here is rotational equilibrium versus gravitational equilibrium. So rotational equilibrium is the ampullae of semicircular canal contain hair cells with stereocilia embedded in cupula. When the head rotates, the cupula is displaced, bending the stereocilia. Thereafter, nerve impulses travel in the vestibular nerve to the brain. Gravitational equilibrium, the urticle and the saccule contain the cell within the bend toward the cuticle. The largest of its stereocilia, nerve impulses increase in the vestibular nerve. If the stereocilia bends away from the kinocilium, nerve impulses decrease in the vestibular nerve. The difference tells the brain in which the direction the head is moving. So you can see here, rotational equilibrium is looking, left or looking right, which you can see this dude right here is doing. And then gravitational equilibrium with this lady right here as she looks down or looking up. And that's how it varies on how the mechanoreceptors help with that equilibrium, so it helps the brain keep that homeostasis. And that is it for this video. Bio lecture number 8 is done. Like, subscribe, comment. You guys know what to do. You know the drill. You've been here before. See you later.